Introduction to Microbiology. What is microbiology? Microbiology is the study of living things that are too small to be seen with the naked eye. This would include bacteria, viruses, fungi, and protozoa, which are known as microorganisms. For billions of years, microbes have immensely shaped the development of the Earth's habitats, and the evolution of other life forms. Studies in microbiology have led to greater understanding of many biological findings. Evolution. Evolution is the adaptation of microbes throughout billions of years to live and endure the changes on Earth. These adaptations gain and lose traits through the process to favor survival for organisms and microbes. Different branches of microbiology. Medical microbiology is the study of microbes that cause disease in humans and animals. Public health microbiology monitor and control the spread of diseases in the community. Immunology studies the complex web of protective substances and cells produced in response to infection and cancer. Industrial microbiology safeguards food and water, also includes biotechnology, the use of microbial metabolism to arrive at a desired product. Agricultural microbiology concerned with the relationship between microbes and farm animals. Environmental microbiology studies the effects of microbes in the Earth's diverse habitats. Two different types of microorganisms. Cellular microorganisms consist of bacteria, archaea, protozoa, and fungi. Acellular microorganisms that cause human disease are viruses and prions. All microorganisms cause human disease. Microorganisms reduce rapidly. This is why microbiologists study every aspect of microbes. This includes cell structure, function, genetics, taxonomy, evolutionary history, and how they interact with living and non-living environments. Helminths. Helminths are multicellular animals whose mature form can be seen with the naked eye. An example of helminths include parasitic worms. Helminths fall in the study of microbiology because of the diseases they cause involve microscopic eggs and larvae. Helminths. The top picture shows helminths in their microscopic stage of life. And as pictured, you can see someone using a microscope and taking a picture of the larvae or eggs of the helminths parasitic worms. The bottom shows matured parasitic worms, and the bottom picture shows parasitic worms in their full-grown state or matured state. Eukaryotic and akaryotic microbes. Eukaryotic means true nucleus. Akaryotic means no nucleus. Akaryotic microbes would be bacteria and archaea. They have no nucleus. Bacteria is ubiquitous. I think I'm saying that right. Meaning found everywhere. They may present small size, but are found in millions. Photosynthesis. And this is just a diagram of photosynthesis showing how photosynthesis starts and ends and how the cycle just continues and continues. Photosynthesis is the light-fueled conversion of carbon dioxide to organic material. Bacteria invented photosynthesis long before plants appeared. The process of photosynthesis formed the base of an ecosystem and fueled the next trophic level. Composition. Decomposition is the breakdown of waste or matter into simpler compounds that can be directed back into the cycle of living things. Decomposition is important because it keeps a steady flow of nutrients available in the environment. And this is just a picture or diagram of decomposition, and it also shows photosynthesis, so it shows how it is a continuous cycle of photosynthesis breaking down to decomposition, and then it's respirated back into the air and it goes back into photosynthesis. Human use of microorganisms. Humans purposely manipulate microbes and microorganisms to make products. This is called biotechnology. Recombinant DNA technology is the process of manipulation in the genetics of microbes, plants, and animals. Bioremediation is the process of introducing of microbes into a new environment to restore stability or to clean up toxic waste. Infectious disease. Pathogens are microorganisms that cause harm to the human body. Most microorganisms that encounter the human body do not cause harm. They are usually beneficial to humans. The World Health Organization, or WHO, estimate a total of 10 billion new infections across the world each year. Cell organization. Organelles are small, double-membrane structures that perform specific functions in eukaryotic cells. Examples of organelles are the nucleus, mitochondria, and chloroplast, and a couple other examples would be ribosomes, and trying to think here. Ribosomes would be another one. They all have specific functions that carry out in the cell. Humans are eukaryotic cells. Historical foundations of microbiology. Spontaneous generation being made from air. This just pretty much says that spontaneous generation, spontaneous means spontaneously made. It's not derived off a certain source or a particular source. So spontaneous generations mean spontaneously made. Kind of like falling out of the air. In 1775, John Needman became close to disproving spontaneous generation. He conducted an experiment by boiling liquids. His theory that organisms could not live in extreme heat situations. He boiled his liquid within the flask, then immediately sealed it. The liquid became cloudy, indicating microbe growth. At this time, they did not know that endospores could not be killed with heat. Endospores are asexual spores that develop inside some bacteria. Louis Pasteur in 1859 put that theory of spontaneous generation to rest. He recreated the research, but with different circumstances. He allowed his flask to remain open so that any life form could enter the flask. He invented a swan neck for his flasks, so when microbes fell in by gravity, they would not settle in the bottom of the flask. The microbe settled in the crook. If the neck was intact, no microbes grew. Pasteur found extreme luck in his experiment. Robert Hooke. In the 1660s, Robert Hooke studied various everyday things. He described for the first time cellular structures in tree bark and sketched little structures that seemed to be alive. He did tests on just multiple things, household items, trees, just random things he would swap and do different tests and research on. Robert Hooke invented a single lens microscope himself. The development of medical microbiology. Early experiments on the sources of microorganisms led to the profound realizations that microbes are everywhere. This research led to the immediate applications in medicine. In the 19th century or the 1800s, this led to the use of sterile, aseptic pure culture techniques. Development of aseptic techniques. Robert Koch linked a microscopic organism with a specific disease. Dr. Holmes discovered that women that had home births over hospital births were less likely to set up infection. His research showed that not washing your hands, leaving the morgue, going into the birthing room, it transmitted a lot of microbes, which caused a lot of infections. English surgeon Joseph Lister noticed these observations and took the first step to introducing aseptic techniques. These are just pictures of microorganisms. The first one is bacteria. This is just one that I selected. There is countless other pictures of bacteria. I just selected this one so we could kind of get a clearer picture of exactly what we were looking at under a microscope. The second one is fungi. This shows fungi spores, what it looks like under a microscope. There wasn't many different pictures. They all had kind of the same similarities under a microscope, but there was some different pictures. And then this one is protozoa. This picture shows what protozoa looks like under a microscope. There wasn't many different pictures that I noticed when I looked. They all looked kind of similar in the shape and structure of actual bacteria, but overall there is differences.